Welcome back, dear students. Let's continue with microscopy, especially the types of microscopy. We had previously mentioned about the bright field as well as the dark field microscopy, and now we are going to deal with the phase contrast microscopy. In phase contrast microscopy, as we had mentioned, we convert slight differences in the refractive index and the cell density into easily detected variations in light intensity. And what happens is the condenser of the phase contrast microscope has an annular stop, an opaque disc with a thin transparent ring which produces a hollow cone of light. And as this cone of light passes through the cell, some light rays are bent due to the variations in the density and the refractive index within the specimen and are retarded by about 1/4 of the wavelength. So if you're going to take a particular specimen as such, there would be what happens, slight differences in the refractive index as well as the cell density will be different at different locations. And as a result, what happens, the light intensity with or the variations will be brought about in the light intensity. Okay, the light will be bent at different angles. And you can see that sometimes the light would also be retarded by 1/4 of the wavelength, and the light which we put onto the specimen is also a hollow cone of light. So we'll see how this hollow cone of light is being created and how the light is being retarded by 1/4 wavelength in order to attain high contrast, which was being obtained in phase contrast microscopy. Coming to the history of phase contrast microscopy, it was being invented by Fritz Zernike, and he was being in 1953 he was awarded the Nobel Prize. It was in early 1930s that he found out this microscope, and it enables us to visualize live cells as well as cellular processes can be viewed under the phase contrast microscopy. Hope you remember that I had previously mentioned about the use. I personally had used phase contrast microscopy to view the budding yeast cells. So the living cells, even when they are not being stained, can be viewed under a phase contrast microscopy. And small phase changes in the light rays induced by differences in the thickness as well as the refractive index of the different parts of the object, they are being transferred into differences of brightness or the light intensities. It doesn't mean that the whole thing becomes dark or anything, but the intensity of the brightness will be varied at different parts of the specimen, and as a result you get a better contrast. And in simple terms, if we have to say, phase contrast microscopy is the translation of invisible phase shifts into visible differences in the intensities. And the phase changes are not detectable to the human eye, whereas the brightness or the light intensity can be detected by the human eyes. Now, what is a phase change and all that? We are coming to that. But before that, let's see what produces a hollow cone of light in a phase contrast microscope. Now, in the case of a phase contrast microscopy, two different adaptations are there. One is the presence of an annular diaphragm, and the second one is the presence of a phase plate, which is being needed. These two things are needed for the phase contrast microscope. The substage annular diaphragm, it is mainly being located below the substage condenser. Okay, and the substage annular diaphragm helps to create a narrow hollow cone of cone or a ring of light to illuminate the particular object. So now this is a, if you go to look, this is a condenser lens, and what below this condenser lens you have the annular diaphragm. This is the upper side view and this is a lower side view. The another thing that we have is when you look into the microscope, sometimes you might have seen only like this. You are not looking into the details of it, but on the other side, on the lower side, you have this annular diaphragm over here. We will see what is it. The diaphragm is in such a way that you will allow only a hollow cone or ring of light is being coming from the is falling onto the specimen. Now, in addition to the annular diaphragm, we also have a phase plate. The phase plate is also called the diffraction plate or the phase retardation plate, and it is located at the back focal plane of the objective lens. And the phase retarding components are coated on this particular plate. And what happens? It is a transparent glass disc with one or few channels, and the channel is coated with a material that can absorb the light but cannot retard it. And the other portion of the faceplate is coated with a light retarding material called the magnesium fluoride. And the faceplate helps to reduce the phase of the incident light. So if you look into this pictorial representation, you can see that uh, if this is the condenser, on top of that we have a phase annulus, and this is a specimen, and from the specimen the light will pass through a phase plate, and the phase plate is coated with some material so that it will be retarding the light to some angle, to some extent. So now coming to look at these two rings. If you go to look the first one, the phase annulus. The phase annulus is coated with some materials so that the total area here is being found to be what? It is not permitting light. But here there is an area, or there's only a ring of it, so that a hollow cone of light is being prepared. Now where is this? So this is a phase annulus, or referred to as a, we had mentioned in the previous act, like the annular diaphragm or the phase annulus, that is mainly being found towards the where, on just close towards the condenser, just below the condenser lens. So you have the condenser lens over here, and just below that you have the phase annulus, and that will produce a hollow cone of light. This hollow cone of light then will further pass through the specimen, and from the specimen the light which has been passing through that will pass through a phase plate. And the phase plate, as we had mentioned over here, it is being coated with a what? A light retarding material. What is that? Do you remember that? That is magnesium fluoride. So the phase plate is coated with what? Magnesium fluoride, so that the light which is being passing from this specimen will get retarded to some extent. Okay, and if you go to look into the phase plate, now you see this, there are portions of coated with light retarding material, and there is a portion devoid of the light retarding material also. So based upon which portion of this the light from the specimen passes, what happens? Changes in the wavelength of the light will happen. Okay, and that would lead into the contrast. Now what is that? We'll just have a look at it. Okay, so if you go to look the unstained cells, they cannot create contrast under the normal microscope, but when the light is being passed through the unstained specimens, it encounters regions in the cells with different refractive index and thickness. And so when light rays pass through the area of high refractive index, it deviates from its normal path, and such light will experience a phase change or a phase retardation. And the light passes through the area of less refractive index remain undeviated. So if the refractive index of the specimen is being found to be higher, what happens? The light will get deviated. And if it is not, it's of less refractive index, what will happen? No phase change will happen, or it will remain undeviated. And the differences in these phases between the retarded and the unretarded light rays is about 1/4 of the original wavelength. And if you go to, like, normally if you look, when if it is a refracted and a, what is it, if it's a retarded as well as an unretarded one, what happens? They will have almost 1/4 the wavelength of lambda by 4, yeah, 1/4 lambda of wavelength different will be there. So what happens? And these minute changes, normally it is not being visible using our normal eye, but when you're using a phase contrast microscopy, we are using certain plates called the phase plates, so that this wavelength, this small wavelength changes also can be amplified, and we could be able to view it in a better way, and we get a better contrast. Now let's just look at this. Now if you go to look what is I'll just tell you how is the difference between a wavelength. Okay. Now consider two different points on a particular specimen. Consider A and B. In if one specimen, if this the point B has a light has been passing through it and it is having of less refractive index, okay, sorry, it is having a higher refractive index. Consider such case, okay. If B is having higher refractive index, what happens? You just look at this wavelength. It is almost half a wavelength retarded. Now just look. A wave is what? When you consider this till here, it becomes one wave. Okay, from here till here. But when A reaches the wavelength, the part when the light passes through A, the wave is moving at this intensity. That is not the height of the thing. The start of the wave starts like this and it ends here. So, but you look, the light waves which have been coming from B is being found to be slower. Just look at this. Almost half wavelength difference is there between these points A and B. That is, the light coming from the point A and the light coming from point B is being found to be less. Now, not less, they are being found to be, there's a small phase change between them. If you consider 1, 2, 3, 4, okay, these are the 4 plus 4.5 of the wave is being present here. And here also 4.5 of this is there. But if you go to look at the light which is being coming from the point A, it is reaching fast, okay, and the ray which is being coming from the point B is half wavelength retarded, or it is coming slower. So what happens? The crust and the troughs. Now this is a trough and this is a crest. The crest and the troughs will cancel with each other. So what happens? The lights coming from these two lights together come and it becomes it results in a negative or a destructive interference, or there would be a less brightness from those lights which have been coming from A and B. So in the regions where the lights coming from A and B meet together, there will be less brightness. But if you go to consider this case, okay, if there are 2 points in such a way that the light which has been passing through those specimens are of the wavelengths are in the found to be in same phase. Now here they are not being found to be in same phase because the crest and crest is not meeting and the trough and trough is not meeting. But here you can see the crest and the crest, both the crests are meeting. That means they are in phase. Here also the crest is meeting, crest is meeting. So they are being found to be in phase. So at those points where the light emerging from them are being found to be in the same phase, or if 2 points are having the same refractive index, what will happen? The light which is being coming from these 2 points will be in phase, and as a result a constructive interference would happen, or they would combine together and the intensity of the wave will be increased. So at those regions what happens? They will appear to be brighter. So depending upon the different specimens, okay, depending upon the refractive indexes of different areas or different points of the particular specimen, the light which passes through them will be retarded, or sometimes they would be in phase. If they are being retarded, what happens? When the light associated from the closely located points would either undergo negative interference if they are not in phase. You just understand what is in phase, what is it, out of phase and in phase. If they are in same phase, what happens? The lights which have been coming from the 2 points would be like crest and crust. This is the trough and here also is the trough. So it would result in, they will combine together and you will get a constructive interference. And in the case of those regions where what happens? There is a change in refractive index, there would be phase changes. And so what happens? A negative interference or a destructive interference would result, or less, or those regions will appear to be as less brighter in nature. Now let's see how such differences in the wavelength is being created. So as I told you here, if there is a, if there is a change in the wavelength of the light, or the phase shift is there, that will be amplified. Normally, if you go to look into a specimen, okay, if you have a bacterium over here and there is a light which has been passing through it, in a phase contrast microscopy, those portions of those light which has been passing through the bacterium, which will be deviated, they would be only being deviated by 1/4 of the wavelength, okay, or they would be only 1/4 out of phase. But what happens is, when those light which has been passing through this region, okay, now this is a phase plate which has been present in a what is this? This is a phase plate which has been used in the phase contrast microscopy. So those regions, if those light which has been passing through this area, will be again deviated by 1/4 the wavelength. So totally it will be half wavelength out of phase. So when it passes through the specimen itself, it will be 1/4 wavelength. And if you go to look such differences, normally we won't be able to view it, or such small variations in the, or for such small phase shifts will not be visible to our naked eye. So what Zernica did is, he placed a phase plate, as I told you, which is earlier, the phase ring. You hope so you remember the phase plate. Okay, the phase plate is in the center. It has what? It is being coated with a what? A wavelength retarding material. Okay, so what will happen when the light passes through the center portion? What happens? It gets again becomes delayed. So when it passes through this again, when it is passing through the specimen, it is 1/4 wavelength out of phase. But again, when it passes through the phase plate, what will happen? Again 1/4, again it is being delayed. So in a result, the light which has been coming from the specimen or from some regions, they will be 1/2 wavelength out of phase. And those which have been passing through this, okay, they will be in, there would be a difference between them. And the deviated and the undeviated rays will cancel with each other. So that's what we have showed here. The, what is it? This is the thing what we showed here, negative of the destructive interference. So at some regions there would be negative or destructive interference, and in some regions there will be, as we had told here, there would be positive or constructive interference. So depending upon the change in the refractive index of the particular or different parts of the specimens, negative interference or destructive interference would result. So when the unretarded and the retarded light is combined together, you get a negative or a destructive interference, because the crest and the trough of the unretarded and the retarded light rays will cancel with each other. But with destructive interference, the image of the specimen appears to be darker against the bright background. And on the other hand, if the undeviated light is passing through the phase retarding material, the two rays will be in the same phase sometimes, and sometimes, okay, if it has been passed in a phase retarding material, okay, sometimes they would be in the same phase. Like, depends, at some regions they would be in phase and some regions they would be in out of phase. If they have been found to be in the same phase, what will happen? It will be in a positive and a constructive interference. So in background, what happens in the constructive interference, the image of the specimen becomes brighter against a darker background. Thus, in phase contrast microscopy, the combination of destructive and constructive interference will create a high contrast in the final image. Now, this is the high contrast, as we can see. If this is a bright field microscopy, you can see it only like this. But since small phase shifts here are also being amplified and being detected using the phase plate, what happens? Amplified, or it is being changed by the small phase changes are being shifted because using a phase plate. What happens? High contrast happens in a phase contrast microscopy. Now, what is the use of phase contrast microscopy? It enables us to visualize unstained cells, and even cell organelles such as mitochondria, nucleus, as well as vacuoles can be seen. And sometimes cellular events such as cell division, phagocytosis, cyclosis, etc. Cyclosis, these are all can be seen. And cellular movements such as the chromosomal and flagellar movements can be viewed. The cytoskeleton can be studied, the cell membrane permeability, and also for the to observe living cells in the tissue culture and to monitor their growth also we can use it. Now, it also has some disadvantage because we know only thin layer of cells can be viewed under the phase contrast microscopy, and sometimes you can see that when you view certain images or certain specimens under the microscope, phase contrast microscope, a bright halo might also be found, and the formation of this halo is due to the partial or the incomplete separation of the direct or the intermediate. You can see this, a small halo is also being found to be formed. So let me just conclude over here. You have a annular stop over here, so that a hollow cone of light is being created, and then it passes through the specimen, and from the specimen it passes through the what face plate, and the direct rays are advanced by 1/4 length as they pass through the phase ring. Okay, and most diffracted rays of light pass through the phase plate unchanged because they will miss the phase ring, and the diffracted rays are retarded by 1/4 length after passing through the objects. So the direct rays will be advanced by 1/4. So what will happen? In effect, they will be 1/2 or half the wavelength difference will be there, and both constructive and destructive interference will result, and you will get some regions which are being found to be brighter and some regions which are being found to be less bright. So a high contrast is being obtained. Then one more thing, we'll continue with the differential interference contrast microscope. Is here you can create image by detecting differences in the refractive index and thickness of different parts of the specimen, and it is also an excellent way to observe living cells. And here two beams of plane polarized light at right angles to each other are used to generate the light source, and you can see that the object beam passes through the specimen while the reference beam passes through a clear area of the slide, and after passing through the specimen the two beams are combined and they interfere with each other to form an image. And so live unstained specimen can be viewed bright, it appears to be brightly colored and seems to be pop out from the background, giving the viewer the sense of a 3D image. Now you can use cell walls, endospores, granules, vacuoles and nuclei, etc. Now this is the case of a differential interference contrast microscopy. So what is the difference in this case? Here also interference plays an important role, but you are using two beams of plane polarized lights which are being at right angles to each other, and when they pass through these specimens, of course a differential interference happens and you get a 3D view of the specimen. This is a micrograph of the protozoan amoeba proteus, and it appears to be 3D in nature. Hope so it is clear to you right now. With this we come to an end of phase contrast microscopy as well as the differential interference contrast microscopy. Thank you for your patience.